Welcome back, friends. In the last part of this lecture series, we talked about the mechanism of the co-translational translocation of the proteins to the ER. Okay, and we managed to open this channel, which is also called as transloco. Okay, initially the ribosome and the mRNA, okay, and this nascent peptide was attached to SRP and the SRP receptor, SRP particle and the SRP receptor. But now these are released, and after this our channel is opened, and now the protein which is elongating, it can pass through this channel. But the question here is what we are going to do with this elongating, or the protein which, if it has synthesized fully, what we would do with this protein. So there are two scenarios. First is, if the protein is still in synthesis, if it is still being synthesized, protein being synthesized. The second case is, if the translation of the protein or the protein synthesis has been completed, translation completed, or we can say after the protein synthesis is completed and now it is present in the ER lumen, if it's a soluble protein, and if it's a membrane protein, it is now present in the ER membrane. So both these scenarios, the protein in these cases, it is subjected to various modifications or the processings which are known as quality control of the protein, and it mainly happens or occur in ER and Golgi. Now here we are mainly concerned or interested in checking the quality of the protein by giving it various treatments, which are, we can say, the modifications or the changes that change the chemical nature of the protein, or we can say the protein chemistry is changed. So we are mainly dealing here with the modifications of the protein, and this is happening when the protein is being synthesized and after it is synthesized, that is fully formed. Now what is the definition of this modification? In scientific words, we say any enzyme catalyzed change which is done on the amino acid sequence of the protein, either by removing few or some of the amino acids or by the addition of some functional groups. Now what does this mean? Suppose this was the protein which is being synthesized, or it has been synthesized fully. Some of the sequence of the amino acids of this protein has been cleaved. This is known as signal sequence removal mainly, and we would discuss about this in detail in future classes, okay? Or we can say there is addition of some functional groups. Those functional groups can be alkyl group, methyl group, hydroxyl group, phosphoryl group, something like that, okay? Actually the introduction to this topic is too big, and we are not going into the detailed part of this introduction to protein modification. We are only concerned with few very important modifications which are done to the proteins in the ER, okay? So initially I told you there are two scenarios when the protein is in synthesis process, being synthesized, and after it is formed, synthesis. So being synthesized means The modifications are done on the protein when it's still in the synthesis process, or we can say it is co-translational modification. Okay, modification and translation process is occurring simultaneously, and on the other hand here after synthesis means post translational modification. So we can say there are two types of modification: before and after the protein is synthesized. In this case, here we can say it is the folding of the protein, folding, or it is said as protein folding. One more important modification is the disulfide bonds. Post-translational modification, here we have very important one is the glycosylation. That is the enzymatic addition of the sugar groups to the protein, and then we have glycation, which is the non-enzymatic addition of the carbohydrate or the sugar groups to the protein. Alkylation, addition of the alkyl groups. Methylation. Then we have phosphorylation. We also have granulation. What more we have? Hydroxylation. These all are the modifications, and there are many more, but we are mainly concerned with the protein folding, disulfide bonds, glycosylation. Yes, these things. Now the most important question here is, why do we need modification? Why just we can't use unmodified protein? So there are many reasons, but we will discuss only the important ones. First is one step to next step, amplification. There is nothing like this phrase, I just made it on my own. It means amplification, we know, increase. Okay, so there is an increase in the number of the molecules in the next step from the first step, or we can say the already present molecules are now more in number in the next level. So if we talk in terms of the protein or the genes, that means genes are present on the DNA, and DNA is converted into RNA, and from RNA we get our proteins. Okay, the number of the RNA molecules are suppose 5. Okay, and after splicing we are getting 5 in number. Okay, one RNA is giving one protein, but if we subject it to alternate splicing, one RNA molecule would give to more than one protein molecule. That means we can say 5 is giving to 25 protein. But these 25 proteins are not enough to carry out many functions of the cells. Cell is burdened with too much hard work. Okay, there are too many functions which cannot be carried by these 25 proteins. Actually there is not 25 proteins, I am just assuming this thing. Okay, so what we do is we modify them, we make changes in them by giving some chemical groups or removing some of the amino acids. Okay, I just explained that. Okay, by doing this we are increasing the number of 25 to many more. So in this way we can say indirectly what we are doing, ensuring protein diversity. The second important reason here is the protein survival. Protein survival. And we can say to make the protein fully functional, so that the protein does not lose its functionality. Now to understand this concept, we compare it to something. Suppose there is a baby, human baby, when he is born. Okay, and now he is in adult form. To reach this adult phase, the baby needs to pass this journey. Okay, in order to do this, he has to go through a lot of obstacles and problems. First, he needs parental care till 5. Okay, and after 5 he is sent into school. Okay, and in school the teachers make him learn a lot of things, and after 10 or 12 he knows the difference between what is bad and what is good, and after 20 he is well aware that what he has to do and what he has not to do, what is advantageous for him and what can cause him harm. Okay, what is disaster, he knows it very well that what is disastrous for him. So to be a very successful person, like a doctor, an engineer or a businessman, he needs to know the difference between good and bad. So these are all changes which are really necessary to be a successful person, to be a fully functioning body. Same is the case with these proteins. When they are formed in the ER, they are totally new in the world of cell. They are not aware of what is going on and what they have to do. Okay, in order to survive they need a lot of changes. Okay, suppose if the disulfide bonds are not added, the protein would not be stable, and the protein folding, if it is not done to the protein, what would happen? The protein function would not be executed. Okay, so that is why we need modification. Another very important reason is targeting of the proteins to various subcellular locations or compartments or destinations, whatever you say. Till now, after the synthesis, the proteins are containing ER signal. Okay, they do not know their identity. They are all same, but they are not for one location. We have to send them to many different destinations or the subcellular locations, which are the cellular organelles or the PM or the extracellular matrix. Okay, so in order to do that, we need these changes because they are all same. If we do not do these modifications or the changes, they will not go anywhere. because they do not know where to go. That is the thing. So this was just the introduction part to the protein modification. In the future videos, we will be talking about the detailed mechanism of all these modification types which we have just discussed. Okay, if you liked my efforts, please subscribe to my channel. Thank you.